In this lecture, I will give an introduction to LINX L1000 data, the gene expression data generated by L1000 technology. This is the background in which LINX L1000 project comes into being. First, gene expression signatures are most informative of global cellular state. The cellular state could be disease, physiology, or drug-induced. Second, the connective map project is potentially transformative. I think most people should have heard about the connective map or the CMAP project published in 2006 by the Broad Institute. They applied 164 small molecules, a few cell lines, and mirrored the gene expression profiles using Affymetrix array technology. They are able to connect these gene expression signatures of chemical perturbed cells to that of disease and physiology, and thus provides a systematic approach to find connections between drug and disease. Obviously, the more gene expression signatures we have, the more resources we can utilize to find meaningful connections for our own research. But common gene expression profile methods are expensive and are not scalable to be performed in high throughput, like the Affymetrix array technology used in the CMAP project. So LINX L1000 project is launched as part of the LINX project to generate a large number of gene expression signatures using L1000 technology. The LINX project produces the largest set of perturbations to cell lines, but still covering a small fraction of all possibilities. So what is LINX L1000 data and how the L1000 technology reduces the cost? As I mentioned, LINX L1000 data is generated by the L1000 technology. The L1000 technology mirrors 978 genes in each experiment and estimates the rest of transcriptome, the 22,000 genes, using a model built from GEO. So the reason that the L1000 technology is cost-effective is that it only mirrors 978 genes instead of tens of thousands of genes in common method. Thus, the L1000 technology is suitable for gene expression measurements in high throughput. So why 978 genes are enough to reasonably represent the whole transcriptome? The short answer is gene expressions are correlated. You only need to know one to know the other. To demonstrate this idea, the CMAP team compared the connections between all the available gene expression data on GEO and found only 978 carefully picked genes are necessary to recover 80% of the connections. The criteria for the landmark genes are minimally redundant, widely expressed in different cellular contexts, and contains inferential values. This slide is an overview of the protocol of how the landmark genes are measured. First, mRNA is reversely transcribed into cDNA. Then, landmark gene-specific upstream and downstream probes are annealed to the cDNA and ligated. The upstream probe has a unique barcode sequence. In the next step, the probes are amplified using PCR and hybridized to the beads by their barcodes. Each bead recognizes two barcodes. The beads are then sent to Luminex detectors to measure how many probes are hybridized. The previous slide is the protocol of how a sample is measured in each well. Hundreds of experiments are measured simultaneously on a 384 well plate. This slide shows the common experimental setup. About 360 experiments are normally measured together in a batch. Each experiment has 2 to 4 replicates. The figure shows a batch of experiments with 3 replicates. The red circle are control replicates and the blue are experiment replicates. The replicates of the same experiment are placed in the same well on separate plates. So the number of plates equal to the number of replicates in a batch. There are normally 18 controls replicates per plate. The field name for batch is called brew prefix in the LINX L1000 metadata. So this slide shows the data levels of the LINX L1000 data. So the level 1 is the raw unprocessed flow cytometry data from Luminex scanners. One LXB file is generated for each well of a 384 well plate, and each file contains a fluorescence intensity value for every observed analyte in the well. And the level 2 is in GEX type, and the gene expression value, they are gene expression values per 1,000 genes after deconvolution from Luminex beads. Level 3 is the quantum normalized data, gene expression profile, both directly mirror the landmark transcripts plus imputed genes, normalized using invariant set scaling followed by quantile normalization, first within plate and then across replicate plates, which mean plates in the same batch. Level 4 are the Z-score data. Profiles, they are profiles of differentially expressed genes computed by robust Z-scores for each profile relatively to population control. And the level 5 are moderated Z-scores, which are computed in broad and also characteristic directions by our lab. They are signatures computed from replicate profiles. I think it is useful to have an in-depth analysis on the LINX L1000 ID system. The IDs reflect the experiment setup and is linked between data and metadata. This slide shows the distill ID, which is the ID for replicated gene expression profile. Since both level 3 and level 4 data are on replicate level, it is this ID that are used as the index for the level 3 and the level 4 data. The distill ID consists of brew prefix, plate index, and a well index. Brew prefix equals to batch as previously mentioned. Each batch name is in turn made up of 3 parts, the perturbation group, cell line, and the time point, which indicates experiments in the same batch have the same cell line and time point. Perturbation group is just a broader group concept, consists of several related batches. Plate index is the index of a plate used in a batch. Together with the brew prefix, they make up the unique identifier for plate. Well index, it designates the position where the replicate is profiled. Replicates of the same experiment normally have the same well index. You can see the still ID exactly mirrors the experiment setup. This slide shows the SIG ID, which is the ID for the level 5 data. SIG ID also consists of 3 parts: brew prefix, per ID, and per dose. Per ID is the compound index of the broad institute, as BRD, the abbreviation for broad. In the LINX L1000 data, each experiment is determined by 4 pieces of information: perturbation, cell line, dose, and time points. You can see SIG ID reflects the 4 essentials. Together with perturbation group, they uniquely identify a level 5 gene expression signature. And sometimes the per ID is the per MFC ID, which is the ID for the drug plus the batch information, because they collect the drugs at different times and from different vendors. So they use this per MFC ID to more accurately describe the source of the drug. If you are an experienced gene expression analyst, you probably have wondered why there is level 4 data. Most differential expression analysis methods start with the normalized data and yield differential expression signatures, namely jumping from level 3 to level 5 with no intermediary level 4 step. The reason for having a level 4 data here is that there is a strong plate batch effect in level 3 data as shown in the figure. The figure is the PCA plot of example experimental replicates and control replicates in level 3 data with the same conditions across plates in the same batch. The brown, red, pink, and blue dots are control replicates from 4 plates, and the light yellow dots are experimental replicates of taurine perturbation from 4 plates. Normally, we should expect control replicates are grouped together and experimental replicates are grouped together. But here, the replicates of the same plate are grouped together, which makes no biological sense and is surely an artifact. So level 4 DCIC data are calculated to correct this plate bias effect. The level 5 characteristic direction data are directly computed from level 3 normalized data. How does it overcome the plate batch effect? If you carefully observe the figure, you can find that the control replicates point to the experimental replicate in the same directions in each plate, which reflects the biology of how the experimental replicate is systematically deviated from the control. The characteristic direction approach computes the 4 directions and average them to get the level 5 characteristic direction signatures. The details about this approach can be found in the following YouTube video link. So now let's see what perturbation and cell line we have in the LINX L1000 data. There are more than 20,000 small molecule compounds, in which there are 1,300 FDA-approved drugs, about 5,585 bioactive tool compounds, and more than 2,000 screen hits. There are also 22,000 genetic constructs for knocking down genes or overexpression genes. They consist of 900 targets or pathways of FDA-approved drugs, 600 candidate disease genes, and more than 500 community nominations, which are the genes that are interesting to the biology community in general. The datasets covered more than 18 cells, including primary cells, cancer cell lines cells, stem cell lines, and differentiated cell lines from different tissue types. So here we arrive to the question, I think that is probably most important to your audience. Where to find the LINX L1000 data? There are 3 locations to find and download the data. They are LINX Cloud, Geo, and Maya Lab website. The LINX Cloud hosts about 95% of the data from level 1 to level 4 with the chemical perturbations, gene knockdowns, and the gene overexpression perturbations. The GSE 70138 holds about 5% of the data. They are the LGP05 to LGP09 perturbation group datasets, and it consists of only chemical perturbations. You can find them on GEO with this GSE 70138 index. The Maya Lab website provides level 5 characteristic direction signatures only, and the signatures are computed from data in both of the above sources. In the next few slides, I will show you how to download and analyze data and metadata from LINX Cloud. It is big data and a little bit complex. You have to first register an account to access the website. Entering into the website, there are 4 icons on the upper left corner. The first icon provides access to several apps that help users to interact with the data. They are easy to use and will not be covered in this presentation. The second gear icon offers functionalities to download and analyze the data and metadata. The first API icon is the HTTP service to search and query metadata and is actively updated. The download data icon enables the user to download level 3 and level 4 data and associated metadata. The code icon provides code in various programming languages to parse and analyze the downloaded data. The phase-array function enables the user to analyze the data on cloud without downloading. Notice that the metadata accessible through the API is generally more complete and accurate than the directly downloaded metadata. This slide shows the services provided by the Lynx Cloud API. After downloading the data, you will get basically a matrix with raw IDs and column IDs. The raw IDs are probe IDs for each gene and can be queried through the gene info service. The column ID for the level 3 and level 4 data is the distill ID and can be queried through the instant info service. The column ID for level 5 data is SIG ID and can be queried through the SIG info. Yes, you probably realize that although moderated ZSCORE or level 5 data are not available for download, you can browse and search its metadata using the SIG info service. Here is the workflow for processing data downloaded from Lynx Cloud. This workflow uses level 3 data as an example, but also applicable to level 4 data. First, download the big matrix onto your computer. Then download the code of your preferred language and use the parse GCTX function to parse the big matrix file with the annotate only option set to true. The result will be a list of column IDs, the CID, and raw IDs, the RID. Query the raw IDs to the gene info API to get the information of each gene. Query the instant info to find the column IDs of gene expression profiles that are interesting to your project. Then use parse GCTX function again with the CID option set to the selected column ID queried from the API. The matrix will be sliced according to the selected column IDs and output a submatrix that contains only the data of your interest. Notice that by default, the parse GCTX function will try to parse the whole matrix into your memory. This is not practical since it requires hundreds of gigabytes of memory available. That's why we need to slice the matrix by column IDs to analyze the data. The Lynx L1000 data on GEO is much easier to handle. The GEO page listed several files to download, covering data from level 2 to level 4. Both data and metadata are saved in a single file, and there is no need to query a separate API. The files are in GCT format, which is a simplified version of GCTX format. You can still use the parse GCTX function to parse the GCT files. As you can see, there are two level 4 files. The ZSCORE in one is computed relatively to population background, and the ZSCORE in the other is computed relatively to control vehicles. Generally, I think the broad institute will prefer the file computed relative to population background. If you want to use the level 5 characteristic direction signatures processed in our lab, you need to install MongoDB. MongoDB is the most popular non-SQL database that saves data as objects rather than rows in tables. In this L1000 database, each signature is represented as an object with data and metadata as attributes. The files downloaded from our web page are MongoDB files that can be directly used in the database. Instructions of how to set up is provided on the web page. The only thing that requires your attention is that the database does not store genes metadata. The genes metadata needs to be downloaded separately in 3 files. Lynx Cloud RRD.json is an array of prop IDs, matching the order of genes in the full characteristic direction in Lynx Cloud collection. The order of landmark genes in this array matches the order of landmark genes in the landmark characteristic direction. The GSE 70138 RAD JSON is an array of probabilities matching the order of the genes in the GSE 70138 collection. And the API raw meta JSON file is the metadata information for each probe ID downloaded from the broad API and can be used to convert probe IDs to gene symbols and also be used to determine if a probe ID is a landmark gene. Here are some apps developed in the BD2K links DCIC that uses links L1000 data. Life is a search engine developed in University of Miami that integrates all links content, leveraging a semantic knowledge model and common links metadata standards. iLinks is a computational biology app developed in University of Cincinnati that aims to provide statistical methods and computation tools for integrative analysis of the data produced by the links program. L1000 CDS Square is a search engine developed in our lab to search for level 5 characteristic direction signatures that either mimic or reverse users' input signature. Leech is a metadata search engine for links L1000 data deposited on GEO and provides customized download of level 3 and level 5 data. GEO2 Enricher is a Chrome and Firefox extension that help users extract signatures from studies deposited on GEO. Although it does not directly use linked L1000 data, GEO2 Enricher piped the GEO signatures to L1000 CD² to search for similar or reverse signatures in links L1000 database. The last slide is a summary of resources that might be helpful for your research with links L1000 data.